So it's my pleasure to represent Work Package 3, the experimental toxicology work package within AURORA, and to share a bit of our results with you on the biological effects of micro nanoplastics. So in Work Package 3, we took a tiered approach. We couldn't test all of the microplastics in all of our different models in a way that it was very comprehensive. So instead of doing them all, we thought we would start with a tier 1 screening. As you can see here, screening and prioritization. We would then pick out the plastic polymers that were causing the most toxicity and continue with more in-depth research on fetal placental toxicity markers, and then go forward to placental perfusion, and then end up with studies looking directly at developmental toxicity in different models. So that's what we did with lots of hills and valleys that we had to go over in the last 5 years. Starting with the screening and prioritization, we used in vitro cell culture models for the screening prioritization. We looked at uptake, transport, and toxicity in placental cell cultures. So the placenta, as you can see here, is a really beautiful tissue that is so important for providing nutrition and metabolism and gas exchange to the developing fetus. The placenta is made up of different kinds of cells. The trophoblast cells are the most important cells. They can be differentiated into so-called syncytio trophoblasts, which are actually the layer of cells between the maternal side and the fetal side that provides nutrients and gas exchange. So we used a very widely used cell line, a placental cell line, called the BWO cells that you can look at the cytotrophoblasts and the syncytialized trophoblasts in order to look at effects of micro nanoplastics. As the project went forward, we also decided to look at even more sensitive cells, the trophoblast stem cells that we were able to obtain. And these stem cells differentiate into trophoblasts and are really early precursors of the trophoblast cells. So once we were going through tier 1, we also decided to look in more detail at different markers of immune system responses, markers of DNA and gene expression, markers of cellular senescence in these cell lines. But also we developed some very more complex in vitro cell cultures that I'll introduce in a moment in order to look at multiple cell types that you would expect in the fetal placental compartment and on steroidogenesis. Placenta is a very important tissue for the developing embryo to get the hormones that it needs from the maternal compartment. For the placental perfusion, you just heard about. Oh, sorry, I'm going the wrong way. We looked at transplacental transfer and different toxicity markers. And for the developmental toxicity, we looked at the zebrafish embryo and C. elegans as very well-known alternative models for developmental tox. And we also looked at the mouse embryonic stem cells. So starting with the screening, when we actually wrote the proposal, we thought that we could get our hands on many, many different micro and nanoplastics to do a huge screening. I think we promised up to 100 different types of plastics that we wanted to screen. Well, very quickly in the beginning of the project, it became very clear that it was almost impossible to get our hands on good reference material to do this kind of toxicity testing. At that point, really, the only commercially available particles that we could get were the polystyrene particles that just about everybody is testing. Thanks to work done in the project that you'll hear more about later, we were able to synthesize and generate many different particles, both pristine and weathered, and we were able to test 33 unique particles, which was really a huge achievement, I think, and I'm very proud that we were able to get our hands on all of these different kinds of particles. Not easy. We looked at different sizes, different shapes. We were able to use 5 different kinds of fluorescent polymers. In order to look at these transplacental transport studies, you need fluorescent particles. And as I mentioned already, looking at different sizes and shapes and going from the very low nanometer sizes up to the 10 micron sizes. So the first results of our screening and prioritization work with these 33 different micro nanoplastics was to look at, in our cell lines, in our BWO cells, and in our trophoblast stem cells, if these different micronanoplastics actually caused acute toxicity, cytotoxicity. Do the cells die? Are their membranes affected? The membrane integrity affected? Do they create oxidative species, which is really well known as for particle toxicity? So here you can see a very nice heat map showing green where we did not see any effects and red where we did see some effects. And in general, we really did not see any cytotoxicity with the exception of polyamide. But we did see some membrane integrity effects, but no oxidative potential effects. So rather subtle effects on toxicity. And I'm happy to say that micronanoplastics don't make our placental cells die. I mean, that would be a really dramatic effect. What we do see, though, in the bottom left-hand corner is that all of these fluorescent particles that we were able to test actually are taken up by the placenta cells, both by the cell line that we used and the trophoblast stem cells. So all of the plastics are taken up. They don't kill the cells with short-term exposure, but they are taken up and they stay in those cells. So we were wondering what will actually happen within the cells if you dose them with these micronanoplastics. So we set out to look at immune pro-inflammatory markers and looked at a different panel of immune markers. What we found is that mainly polyamide caused a reaction and the significant induction of proteins. The ADA protein here is a protein that's really important for the function of white blood cells. So these are important immune cells. We looked at transcriptomic profiling. So how are genes reacting to the presence of these micronanoplastics within the cells? And these gene expression profiling experiments showed very clearly that polyamide, again, resulted in many different differentially expressed genes. So these cells are not dying, but there's something going on either with the production of proteins related to the immune system or with genes that are changing with these acute exposures. Right now, we're doing more analysis to determine what these actual gene pathways are after exposure to polyamide. Going further in the next tier, we wanted to look at not only if we have one type of cell in culture, what would happen, but what would happen if we made a cell culture system that looks a little bit more like the fetal environment. So our colleagues within the VU University in Amsterdam created this very unique triculture system. So in this system, what you can see here is the boxes show actually a membrane. This is a transwell system. And on this membrane, on top of the membrane, they cultured both the placental cells and the umbilical cord cells. Well, that's a co-culture of two different kinds of cells, like you would imagine would be in the fetal maternal compartment during pregnancy. And on the bottom of the membrane, they cultured these H295R cells, which are cells from the adrenal cortex, an important gland that produces hormones during life and, of course, during pregnancy as well. So we could look at the effects of three different cells that you would expect in our bodies at the same time when you expose them to these micronanoplastics. And what the researchers here saw is that there was actual transport. When you add the micronanoplastics to the top, to the placenta cells, they do pass right through these three layers into the basal compartment, and they do show transplantation or transport across these membranes. What the researchers also showed here is that on both sides, both on the side of the placenta cells and the cord cells, and on the basolateral side, that the steroidogenic genes, so these are the genes involved in hormone metabolism, excretion, and production, were all changed slightly. There were slight changes to these steroid hormone levels that are really provocative. Well, what do those mean? What would happen if these changes in the hormones in the placenta over a longer period of time would happen? That's where we need more research. You just heard about the placental perfusion studies in which Lawrence indicated that there was some minor transplacental transfer to the fetal side, especially when the albumin levels are low. And importantly, as we saw with the cell line work, these micro nanoplastics stay in the placenta. And what are they doing then when they stay on the placenta? That's where we need some more research. So to finalize our tier 4 screening, we used some alternative developmental toxicology models. For example, the zebrafish, a very well-known model used in developmental biology. What we did here was to develop a method to actually inject the yolk of the 1-hour-old zebrafish egg, rather than adding the micro nanoplastics to the water where the embryos swim in. We actually injected into the yolk, thinking this would be a more realistic way of mimicking micro nanoplastic exposure in the human fetus. We then looked at how the zebrafish developed up to 5 days or 120 hours post-fertilization. The results indicated that the zebrafish embryos exposed to polystyrene and polyamide all internalized these micro nanoplastics. So the plastics got into the body, were spread throughout the body of the embryo. However, no developmental effects were found. We are now still doing the RNA sequencing and immune analysis at the moment. So to summarize, using our cell culture models, all of our studied micro nanoplastics got into the placenta cells. There was no acute toxicity. We tested these micro nanoplastics up to 72 hours, except for polyamide. So this is a plastic to prioritize for sure. Several of these micro nanoplastics induced membrane damage, no overt oxidative stress. We're looking at the specific proteins and genes that were induced by polyamide exposure. We found limited effects on the serotogenesis in this triculture, very unique model, that we did see that these micro nanoplastics pass through the model. Placental perfusion, you've heard about that, and the developmental models show no acute developmental effects. I did not show the data for the C. elegans work or the mouse embryonic stem cells, but there we also did not find acute developmental effects. So our take-home messages: the micro nanoplastics that we tested were taken up in all of our test models, both in living organisms as in cell culture models. They showed no or minimal acute toxicity with the exception of polyamide, and they showed subtle effects on serotogenesis, gene and protein expression that we need to further unravel to understand the long-term effects of those changes. So what we don't know yet, we don't know the effects of chronic exposure. We don't know the effects of the mixtures. We studied all of these micro nanoplastics individually. We don't know the effects on human models. And there's a lot of other things we don't know. But I'm very, very happy that we were able to do this first and very important research in these early life stage models. So thank you. And I have to really, really thank, not only did we create amazing insights during this project, we also helped train new researchers. Dr. Jeske van Boksel, this is her PhD defense. She got her PhD last December. And Hannah Dusha, who started as a postdoc in our project and is now an assistant professor in Utrecht. So very important work. Thank you very much.